Yeah, thank you. Hi, my name is Andre. I'm a PhD student at the Institute of Applied Quantum Technologies. We are part of the Department of Electrical Engineering. This will be a prominent feature in my talk of the Friedrich Alexander University in Erlangen. Our small team of our chair, we are roughly 2 years old now, comprises to 50% of physicists and 50% of engineers, because our goal is to push the topic of quantum technologies and applications of quantum technologies outside of the physics lab, where it has been stuck for the better part of a century now, into the engineering labs and into industrial applications. And when I talk about quantum technologies, I refer to the second generation quantum technologies. In this case, for example, trapped ions, so individually charged particles that are trapped by an electromagnetic field and contained in an ultra-high vacuum. Superconducting qubits, so the powerhouse of the quantum computing community in the 2020s. Basically engineered circuits made from a superconducting material that are cooled to near absolute zero temperatures, where they start to exhibit these quantum mechanical properties. Quantum dots, which are semiconductor nanocrystals that contain single electrons or holes, sometimes referred to as artificial atoms. Solid-state spin systems that use an intrinsic spin in a point defect within a host crystal, and molecular systems such as cold atoms that are neutral atoms suspended by optical tweezers within an ultra-high vacuum and cooled to temperatures near absolute zero. And while all these have their own applications in the quantum technology community, they all have their advantages and disadvantages. And one big flaw they all share is that in order to perform quantum computing algorithms, or in our case, perform quantum sensing, is that they have a very limited lifetime. So at some point, all information vanishes from the system, and this is the limit not only for quantum computing, but also for quantum sensing, in which time frame we can interact with these systems. And in order to elongate this, the usual approach is to just freeze out any thermal noise. Use a vacuum and a cryogenic temperature to get rid of all thermal fluctuations, and this way make our systems much more stable so that we can keep information for even hours in case of nuclear spins. Now, if we want to talk about real-world applications using a ultra-high vacuum and cryogenic temperatures, kind of defeats the point. So this is where one of these systems, namely solid-state spins, comes in with a major advantage. And the interesting part here is that we still use these spin systems that observe quantum mechanic phenomena, but since they are embedded in this very rigid crystal, it shields them from macroscopic noise and from thermal fluctuations. And this allows these solid-state spin systems to observe coherence times that go into the micro to milliseconds, even at room temperature. And this is the big advantage, because these systems, they don't require a high vacuum, because the spin state is encased in this solid-state material, and it allows us to perform our measurements at room temperature. And the poster child for the solid-state spin systems within the last 2 decades has been the nitrogen vacancy center. I'm sure if someone took a look at this field, you will have come across this big player. And the interesting thing is that it is literally just a substitutional nitrogen atom that is stuck within the carbon lattice and an adjacent vacancy that confines 2 unpaired electrons. And these 2 electrons, they make up our spin system. And this is what we refer to as a nitrogen vacancy center. And this is basically it. This is our sensor. Literally 2 missing carbon atoms within the lattice. And due to this nice crystal host, we are able to interact with it at room temperature. It offers long coherence times, so it can save information long enough for us to read it out. And this is practically what we abuse in this case. It is optically active. We can read it out and initialize it optically. And the diamond is a very robust solid-state host platform that is biocompatible. It does not degrade over time. The NV center is not doesn't suffer from blinking, so it just doesn't disappear out of nowhere, and it is very spectrally stable. And this is the reason why this defect in particular now has nearly half a century of research on its back. From the 1950s, when the first electron spin resonance experiments showed microwave radiation being absorbed by the diamond, which revealed a nitrogen donor in the diamond lattice, which we now know is the most abundant paramagnetic defect in diamond. It took 20 years for researchers to find what is now defined as the NV center by its very distinct emission band after irradiating and annealing and creation of these NV centers. Then another 12 years have to go by until scientists first develop the tools to optically address the spin and manipulate it with electromagnetic radiation, and we call this optically detected magnetic resonance. And this is basically a tool, or the first of a manifold of tools to interact with these spin systems. It lets us read out the energy states within this spin system. Then the end of the 2000s went out with a bang, as the measurement hardware became mature enough to show these experiments on single defect emitters. So at this point, we were able to initialize, read out, and control single defects within the host crystal at room temperature. And I cannot mention this enough, because this is really the only system we know at this point that has so nice properties. And this is the reason why it has gained so much traction within the last 20 years. Then in the mid-2000s, the quantum computing community was able to show some milestones and breakthroughs with the NV center, where they were able to show coherent spin control of a two-qubit quantum gate at room temperature, where they would use a carbon isotope in the vicinity of the NV center to perform these two-qubit operations at room temperature. This is the point at which the quantum computing community thought about, okay, how can we improve this process? How can we keep the NV center more stable? And of course, you can freeze out all thermal noise, as I've mentioned. So they would do all sorts of tricks to elongate the coherence time of the NV center and to get rid of all these fluctuations and environmental noise. And this was around the time when the field of quantum sensing gained traction, as people would start to propose experiments where we don't try to get rid of the environment, but we try to use it. And we use these incredible sensitivities of our vacancy to the environment in order to perform measurements that haven't been possible before. We could imagine, or it has been proposed and it has been experimentally shown more than 15 years ago, that you could insert a nitrogen vacancy into the tip of an atomic force microscope in order to scan across a substrate and detect the signal of a single electron on the surface of the substrate using the NV center as a sensor. And this was nearly 20 years ago now, and since then the community evolved and grew, and at this point we are basically done with theory. You cannot really show any new theory on the NV center. We know all about it, and this is, for the quantum sensing community, especially for engineers like me, it's a nice thing. If you are a physicist and you wanted to get into fundamental research, it's pretty difficult with the NV center. But as I mentioned, it's good because we know all the interactions that the NV center's spin system has with the environment. We know how it interacts with magnetic fields, and this lets us use this high susceptibility to its environment to our advantage. And we can detect magnetic fields using the NV center, so basically this missing carbon atom in the carbon lattice, and we can probe magnetic fields. We can abuse the nice symmetry of the diamond crystal lattice to probe the magnetic field vector. And yeah, we can borrow some schemes and some ideas from our friends in the nuclear magnetic resonance community in order to detect NMR signals from single molecules on the surface of the diamond on a nanometer scale. We know its interaction with temperature. We can measure thermal fluctuations with the NV center, which can create millikelvin sensors, precision sensors that offer this nanoscale resolution that almost 10 years ago, more than 10 years ago now, enabled even biologists, who usually are not found in an optics lab, to use nanodiamonds, which they usually use as a fluorescent marker, in order to insert these nanodiamonds into living cells. And now they're able to probe temperature within the living cell and detect these temperature gradients across the domain of these individual cells. Same with electric fields, though the interaction of the electric field with the NV center is not as trivial. This didn't stop scientists from developing schemes and measurements in order to infer the electric field and even use clever experimental tricks in order to extract the electric field vector. One can even treat it as noise, basically, because it destroys this coherence time of our NV center, and we can infer ion concentrations. For example, in liquid electrolytes, one could imagine a battery with a sensor embedded in its electrolyte, and we can infer the ion concentration within this liquid. Or we could use the nanodiamonds inside cells, which is an active area of research since 10 years from our friends in the Netherlands, that infer free radical concentration. So when a cell is stressed, and they infer the radical concentration from the decline in lifetime of our NV center. We can also make the material guys happy because strain and stress within a crystal is nothing else than shifting electric fields of the surrounding host crystal, and we can sense strain across our host material with the NV center. So all of this happened within the last 15 years, and at this point, it's really hard to make something new, because in the last 10 years, we focused on optimization, and we are focused on new applications of the NV center. And this is an important evolution, because we see the same happening, or we saw the same happening with the transistor and with the LED. It spent a few decades in the physics lab, and at some point, the technology was so mature that it could make the jump into the engineering lab and into the industrial application. And yeah, this is practically where we're at. And now, for the rest of the talk, I would like to give you a hands-on example on what measurements these technologies can enable, on an example of our project of ours in cooperation with the University Hospital in Erlangen. Our colleagues at the hospital, they focus on the migration of cancer cells and understanding the dissemination of the tumor within organic tissue. And in order to get the fundamental understanding, we need to observe what's happening. And as our colleagues are biologists and medical researchers, they need tools in order to do so. They can either use light microscopy, which does offer the lateral resolution that is required to observe these individual cells. But we need to add a few tricks in order to distinguish it from the biological background. We need to use stains and fluorophores that suffer from photobleaching, or we need to alter the cell itself in order to produce these fluorescent proteins. Neither of these options are good for a long-term observation. Additionally, depth resolution becomes really hard with light microscopy, because in order to resolve a signal from within the tissue, we need confocal microscopy and very high laser powers, which interact with the surrounding tissues, which is also nothing we want to do if we want to see what naturally happens to these individual cells. On the other hand, we can use magnetic fields as labels and markers. The de facto gold standard in medical imaging is the MRI. Unfortunately, at the moment, it doesn't offer the spatial resolution and the required measurement times in order to resolve these individual cells within organic tissues. We can, however, make use of the basics, so labeling our cells with some magnetic markers. This is something that we can do, and we could do for quite some time now. And if we are able to label these cells, the only thing that we now need is a sensor that can detect them. And as I mentioned, MRI doesn't really work out. So we turn to the NV center because, as I've mentioned already, it is room temperature capable, which is required for our organic tissue. It offers a nanoscale spatial resolution capable of resolving these individual cells within the tissue. And it offers the high magnetic field sensitivity that is required in order to resolve this faint magnetic field caused by these magnetic markers. So what we need at the end of the day is a magnetic microscope, and this is what we've built. We use the diamond as a mediator between the magnetic signal from the label and the optical domain that allows us this microscopic resolution. And this is something that we've built up. So we bring our cell sample with these magnetically labeled cells in close proximity to our magnetic field sensor, and we perform a measurement. And I try to keep this short to give you an elementary understanding of how these measurements are and how easy they are at the end of the day. We use our diamond with our NV centers implanted on one surface. We excite it with a green laser so that we excite our spin system, which then decays and emits a red photon. And these red photons is what we can detect with our camera. And it's interesting to see because you can actually see the diamond glow red in the experiment. And this red fluorescence light is what contains information about the NV center. Then we add a microwave loop because we need a way in order to manipulate the spin state in order to read out the energy levels contained or the energy levels present at the location of the NV centers. And then we collect all the light and we focus it on a camera sensor. And by using a camera, we can spatially resolve the signal across our diamond, which acts as our magnetic field mediator. Now, what we do is we take this microwave that interacts with the electron spin of the NV center, and we start sweeping it across the energy transitions of this electron spin state. And at each frequency, we take an image with our camera. And at some point, at some distinct frequencies, something beautiful happens, and this is where quantum mechanics comes into play. Due to these distinct energy levels within our spin system, we see certain frequencies where we can see and observe a drop in fluorescence. This is where basically our diamond goes dark. It's what we call a dark state. And this happens when our microwave is exactly resonant with the NV center's spin, and it starts flipping the spin population into the other dark state. And the nice thing is, since we know about the theory of the NV center, we know that the spectral position of these minima, of these dips in the fluorescence signal, is linearly dependent on the magnetic field. It's something called the electron-Zeeman effect. And by now detecting where these resonances are for each pixel of our camera, we can infer a magnetic field for each pixel of this camera. And this basically gives us a magnetic field camera that can see magnetic fields with the microscopic resolution required to resolve individual cells. And we can see a direct bijective relationship between what we observe with classic bright field microscopy and what our electric field microscope can see, magnetic field microscope can see. The interesting thing now is that we are completely irrespective of any tissue surrounding it. So even if we would put a few micrometers of tissue above our cells, we would still be able to see them with our magnetic field microscope, which is where the classical microscopy breaks down. Yeah. Thank you. I hope I could give you not only an idea of how quantum sensors evolved over the last half a century, but also why the NV Center is such a prominent figure in our community. I hope I could give you an elemental understanding of how these measurements work so that maybe we can get some fresh applications that find its way into our community.
Thank you.